Okay, all right, welcome everyone. Here we are at our February meeting. So let's just get right into it. Just a few brief stuff and then we're just going to get to Scott's presentation here. So as everyone knows, every third Thursday, this is pretty much a review just for folks that might be new to these meetings. 2 to 3:30 every time, same Zoom link every time. And we do send out reminders. Krista asked me to put some of her additional folks on the distribution list, so I'll do that. If anyone else has folks that you'd like to have on the distribution list for those reminder emails and such, just let me know. We're supporting groups that are working directly in the Delaware River Watershed Initiative, but also groups that are just in the Delaware Basin, but not directly associated with DRWI. This is via DRWI money and Seesaw as well. DRWI, as most folks know, is a William Penn Foundation funded initiative. Feel free to go to this website if you'd like to check it out. Seesaw is a state of Pennsylvania Growing Greener program. Feel free to go there if you'd like to learn more about that. That is available to groups across Pennsylvania, not just in the Delaware Basin. So the goals for these meetings are simply time and space to just check in and provide some information, provide updates, and you know, discuss any issues that come up, questions, etc. We do provide updates. We've moved to just one presentation per meeting. This time it's going to be a focus topic. Scott's going to be talking about turbidity. Here's the main folks that are facilitating these meetings: myself, Rachel, Krista, and Shannon are the Stroud folks. And then we've got Carol Armstrong and George Seeds, who are Master Watershed Stewards from Chester and Delaware County, who are closely working with Stroud. As I pointed out earlier, the meeting is being recorded, so please just keep yourself muted unless you're going to be talking. Just to point out, these recordings are available here at this website, as well as the presentations. This is our general format for the meetings currently. Today's meeting, pretty simple. We've got Scott's presentation. I've got some updates, which I'm actually going to do after the presentation, just to make sure we're good on time. And we do have a Zoom survey today. Again, just same two questions as usual, which we appreciate folks completing. And then Christine Rogers has will take a few minutes to just follow up with some information from her and Juniper's presentation last month. So just with these meetings, well, with this work in general, EnviroDIY in the Delaware Basin, primary goal of Stroud continues to be to support groups and across the basin and using these stations for their own local purposes. Secondarily, we are doing data analyses internally, and that's what Scott's going to be talking about. Any questions at this point? All good? All right, we will move on to Scott. Scott Ensign is the, oh, I didn't put it in here. Scott is the Assistant Director at the Stroud Center. And he's going to be talking about some work that he and Sarah Damiano primarily have been doing on turbidity.
All right, thanks, Dave.
Yep.
Thanks everybody for joining. We've got a snowstorm. You could be outside having a snowball fight. You could also right now be watching the Mars rover land on Mars on YouTube, but you're here with us to talk about turbidity. So that's great. So thanks for joining. And many of you have turbidity sensors on your monitoring equipment. And there's a lot of, it's a deep subject, turbidity, no pun intended. So this was a chance to kind of talk through a superficial look at what turbidity is and how we can use it for assessing stream health. So let's just start. Before we start talking about turbidity, let's start with sediment. So when we talk about sediment in water, we can talk about two different types of sediment in water. The picture on the left, when you see a storm at flood stage and it looks muddy, what you're looking at is suspended sediment that's in the water column. And it's floating around there in the water column. During big floods, like the picture on the right, those cobbles that are around me in this very dry riverbed, when this riverbed is flowing, those cobbles are rolling along the bottom. So we call that bed load. It's another type of sediment, but that's not what we're going to talk about today. We're going to focus on suspended sediment. Again, the stuff that's in water that makes it look cloudy. And that stuff that we're seeing that makes the water look cloudy is a combination of clay, silt, and sand. And generally for the sites that, that we're dealing with here in the Delaware, it's everything from clays to medium sands. So everything from less than 2 microns, which is clay, up to 0.5 millimeters, a medium sand. So, but when we zoom in, what is that? What is that? What does it actually look like if you were to put a scanning electron microscope underwater during a flood in that cloudy, milky water? What's down there? Well, it's really complicated. It's not just individual grains. It's a combination of different size material. It's also conglomerates and aggregates and flocculent material. And it's not only inorganic stuff, what we would think of as mud, sand, silt, clay, but it's also organic material. So there's little bits of algae in there, there's bacteria in there, there's pieces of leaves and sticks and all kinds of other things. So it's when we say suspended sediment, we can be specific and we can say inorganic suspended sediment, but generally when we're talking in a very general sense like this, we're talking about all the stuff that's in there, and it's the kitchen sink of things that go down rivers. When we talk about measuring suspended sediment, you've probably heard two different words for how we measure sediment. You've probably heard the term suspended sediment concentration. You've also probably heard the word total suspended solids. So these are two different measurement techniques, and there are multiple specific protocols for each of these two types of techniques. And moreover, a lot of times we'll use one of these and we'll tweak it in various ways. In the big picture, I want you to be aware that they are two different things and that they differ primarily in how you collect the sample. So when we talk about suspended sediment concentration, if you were to follow the rule book, you would collect your sample using what we call an isokinetic sampling device. So it's a bottle with a nozzle on it, and the water goes through the nozzle at the same speed that water's going down the river channel. So that's really technical, and you can get into the weeds on this stuff. We could talk for hours about the different methods that are involved here. So that's one of the differences, is that you collect it in a certain fashion. The other primary difference with suspended sediment concentration is that you've collected a known volume and you filter the whole thing. So you dump the whole thing through a filter, you dry the filter, you weigh the filter. On the right-hand side, the difference with total suspended solids is generally you dip a bottle in the stream, you try to, as best you can, capture whatever's going downstream. And then usually what you do with total suspended solids is you'll pour off a bit of that sample into a graduated cylinder to filter a subsample of that bottle. And typically that's what's called total suspended solids. From that point forward, it's very similar. You collect the filter, you dry the filter, you weigh the filter. In both cases, the weight that's left on the filter is either suspended sediment concentration or total suspended solids. The good news is that generally speaking, if the mass of your sediment in your sample is less than 25% sand, these are going to be very similar numbers. But if you're in a really big river during a really big flood and there's a lot of sand in suspension, then your total suspended solids measurement may underestimate the total sediment load, so total sediment concentration. So bottom line, two different techniques. Under most conditions, if there's not a lot of sand, they're going to be similar numbers, but we should be clear about what we're talking about methodologically. Okay, so why measure suspended sediment in the first place? I think we can kind of throw that into three different buckets. We can take measurements of suspended sediment and we can compare them against a regulatory threshold or a biological threshold. Are they above or below a certain value that's we would consider dangerous to something? The second really broad category is that we can use patterns and trends in suspended sediment concentration to understand things like sediment sources, transport, deposition, erosion, broad field of different geomorphic questions. And then third, if we convert that suspended sediment concentration into a load, we'll get to that later. We can, oftentimes we're interested in, is there more sediment entering a stream reach than is leaving, or vice versa? So that's another common question. So three different kinds of questions that we collect suspended sediment data to answer. It's a lot of work. So we talked about methods, suspended sediment concentration, total suspended solids. If you get into the weeds with particularly measuring suspended sediment concentration, it's a lot of work to do it really well. So is there a shortcut? And hinting here at what we're transitioning to talking about is turbidity. No, there's not a shortcut to measuring suspended sediment, but there is turbidity, which is not suspended sediment, but we're going to talk about ways that we can relate one to the other. The great thing about measuring turbidity is that it's cheap, relatively speaking, it's easy, and you can collect data every 5 minutes like many of you do day in and day out with little work actually making those measurements. The downside of turbidity is that the unit of measurement itself doesn't have any meaning. We have to relate it to something. It's a very weird measurement. In fact, there's about a dozen different units that you can apply to a turbidity measurement. You all are generally using what's called an optical backscatter sensor. And the way that works is that it shines light out into the water from the end of the sensor, and it's measuring the light that bounces back at a certain angle back to the sensor. And again, we could talk for another hour of all the different kinds of ways that you build turbidity sensors to measure a very specific type of particle. But let's keep it simple for now and just emphasize that what you're measuring is optical backscatter turbidity, and the company that probably you have a sensor from is Campbell Scientific, and they've done a lot of work and a lot of engineering, and they've told us that that measurement that comes out is in units of nephelometric turbidity units, NTU. So that's one of the 12 different kinds of turbidity units that we're going to talk about is NTU, and that's what you're collecting, or some of you are collecting with your Campbell sensors. Okay, so let's go to this first kind of basket of reasons why we measure turbidity, in this case, going into the turbidity, and it's to compare the values in a stream to either a regulatory threshold or a biological one. So what's happening and why would we care about those different thresholds? So turbidity, as you know, blocks sunlight. So the water gets cloudy during floods or when there's a source of sediment. It blocks sunlight that algae and aquatic plants need. So that's one effect that turbidity can have on streams, and that's one of the potential thresholds that we think about. Another thing that turbidity and the underlying suspended sediment that we're capturing there in a measurement of turbidity is that it's potentially clogging stream bottom habitat, which affects macroinvertebrates. It can dislodge them. It can push them into drift. It can clog their nets. If you're a fish, it can interfere with your vision and how you're feeding. Suspended sediment and our measurements of turbidity are also an indicator of particles, which are basically providing a place for things like phosphorus and bacteria to attach to. So we care about the magnitude of turbidity because it's an indirect indication of how much phosphorus and bacteria might be going downstream. And also, finally, it goes without saying that when we think about the impact of turbidity on streams, we should also consider our own perspective and perception of streams. And there are many studies that have gone into trying to figure out at what point the human eye, looking at a stream, at what level of turbidity will we think that looks like a degraded stream. So there's actually numbers out there that indicate how we perceive streams and what that level of pollution might be. So those were all the kind of the biological, some biological reasons and chemical reasons, ecological reasons that we care. When we think about turbidity in terms of putting a regulatory measurement out there for streams, it gets really complicated. So there's a lot of stuff on the slide. Go first to the state that you're in and read the bullet point or whatever the threshold is for turbidity in your state. And you're going to see two things. First, in most of these, in Pennsylvania and New Jersey, there is not only a magnitude of turbidity, say 15 nephelometric turbidity units, but there's also a time attached to it. And that's really important because when we're looking at the effect on biology, on fish, on bugs, on people, on algae, it's not just how high the turbidity gets, it's how long it stays high. So each state around the country has instituted their own turbidity standards. And in some states, it's more complicated than others. The EPA has not set a nationwide level regulatory limit, and that's why each of the states are free to set their own regulatory limit. However, EPA did provide for each ecoregion around the country, and there's like 12 different ecoregions. They said for your ecoregion, we suggest you consider a reference condition, and they compiled a bunch of data for basically for the Delaware River watershed. EPA's suggested reference condition is 1 to 2 NTUs. So you can keep that in mind as what's a really pristine, clear, beautiful stream. It's 1 to 2 NTU. Okay, so going a little bit, just to emphasize this point on time scale. So if you're a fish in a stream or a macroinvertebrate, the length of time that you have to endure a particular condition can be important as the level of turbidity that you're experiencing. And so, for example, on the right, you've got a really high, very high turbidity in a stream, maybe during a storm that lasts for a day. Fish, macroinvertebrates, in some cases, they're going to be able to get through that, and it's not going to have a long-term effect on them. But you could have the same impact with a much lower turbidity that lasts for hours or days. You can have the same effect. So in the cases that we're going to talk about today, I want you to keep in mind, and when you think about measuring turbidity, keep in mind that it's not just the maximum level of turbidity that happens during an event or during a pollution event. It's how long that event is sustained that has a big impact on the organisms that we're really interested in here. So I dug into your data and I went through the SitSci data that is all on Monitor My Watershed, and that we periodically harvest data from you if it's not up on Monitor My Watershed. And I went through that data and explored it basically to not to really do a strict analysis, but just to explore the magnitude of individual sites, compare those sites, and look at how conditions change between storm events and base flow periods. So there's a lot of data out there. You all have collected millions of data points on turbidity. And so I had to start whittling my way down through that list to a data set that made sense to me, that I could start to wrap my head around. So in six steps here, here's what I did. I selected only the turbidity data from your sites that was within 1 week of a sensor cleaning that you had noted on a field data sheet, because I wanted to make sure that the data that we were looking at was reliable, had been pretty close to a period of time when you cleaned that window on the turbidity sensor, because that makes a big difference. The next step was to exclude outliers. So if there's a point that was hundreds of NTU higher than everything else, we got rid of that. And then from what was left, I did a statistical smoothing to kind of even out the 5 minute to 5 minute high frequency variation there. And then I used a program, an algorithm, to separate the data at each site into events, periods when turbidity spikes up, I call that an event, and periods when turbidity was not spiked upward, we'll call that normal. It's not necessarily related to a storm, but it might be, and I'll show you some examples. And then averaged those normal conditions and took the peak of those events. So I'm going to show you the normal conditions and the peaks. So I'm going to show you that data for a couple of sites, and we'll look at some different scenarios out there that you've collected data on and compare events and normal periods. But we kind of have to have a threshold to compare that against. I showed you various regulatory limits for the different states. That's one way to do it. We could have put dashed lines across the figure here, and I could have presented this to you in that fashion. I think it's more helpful for the conversation and for the sharing that I wanted to do today to kind of put it into perspective of thinking about different thresholds that you might cross with different levels of turbidity. So one of the reasons that turbidity is so difficult to put regulatory limits on is because biology and streams differ wherever across the country. So there are studies out there, and they're specific to a particular place. For example, in the Midwest, a turbidity of 5 NTU was associated with a 50% decrease in macroinvertebrate richness. Does that mean that that applies in Pennsylvania or New Jersey or Delaware? I don't know. And I'm not going to say that it does, only that somewhere we've seen that effect in the literature. Another example, when you get to 12 NTU, brook trout reaction distance when in their visual field shrinks by 50%. So we could look at if turbidity crosses that 12 threshold, gives us an idea that we're starting to infringe on vision of fish. 23 NTU, we have a particular caddisfly species starts to die. Rainbow trout fry grow slower in some experiments. Again, does that mean in your stream that's what's happening? I can't tell you that's true. These are results from the literature that, again, give you some perspective on what these thresholds are. Okay, so let's look at one scenario here. Let's look at Ridley Creek, and you have a group has a site upstream and downstream of an old mildam that's been drained. So you can kind of see Sinclair Lake there. The footprint of it is still obvious. And there's turbidity being measured upstream and downstream of this former site. So this is what the turbidity looks like. So there's 4 diamonds here. All right, so let's look at here on the left. This is the upstream site. And there's a little squiggly down here at the bottom. That's the normal turbidity. It's really low. The interquartile range, half the values are within this tiny little speck here. Very low turbidity. During the events, half the values fall within this diamond. and the median turbidity is at the halfway point of this diamond. The difference between this upstream site on the left, the downstream site on the right, appears to be not so much what's happening during events. What appears very different to me is the fact that the normal conditions in the stream, in other words, not when we have events, but when you have base flow conditions generally, is higher. And it's going into that second tier of threshold, that second color shading of brown here, to give you an idea of that it's starting to have a biological impact according to prior studies. So there's a case study that shows that it's not so much the storms that appear to have the big difference, it's the base flow period. Okay, let's go to Pennypack. Here's an example where we have an upstream and a downstream site that some of you are familiar with. And here, again, comparing the diamonds that are near the bottom, these are the non-event periods, the normal periods, the base flow periods. These at the bottom are more similar than these event peaks, the bigger diamonds showing up here. So in Pennypack, it looks like whatever's happening between these two sites the big difference isn't at normal flow, it's during events when we see higher turbidity downstream. And learning a little bit more about this site, talking to David Bresler, one reason might be that Northampton Creek enters Pennypack between these two sites, and Northampton Creek has a sediment TMDL. Perhaps there's a source here. So I'm showing you this example just as a way to demonstrate that when you start analyzing turbidity data and grouping it together like this, where you're looking at upstream and downstream effects, you can start to get an indication of where problems might originate, and it helps you develop hypotheses about what to investigate further. Let's look at one more. This is Cherry Creek. And again, here's a situation in which the diamonds on the bottom and the diamonds during the events are all increasing from upstream to downstream. So what's going on here? I don't know. But it's a trend that you can find in the turbidity data that suggests that there's something going on here and it's worth exploring in more detail. Finally, thinking about trout streams, because we were looking at thresholds for trout. Let's compare 3 trout streams, Paradise, Broadhead, and West Portal, that have trout populations, wild trout, to a reference stream, which is Aquashicola on the far left. And we see that all of the trout streams, Paradise, Broadhead, and West Portal, all have very low non-event average condition, turbidity condition, but that the peaks during events are definitely higher than Aquashicola, a reference stream. Does that necessarily mean that trout are challenged by turbidity in these environments? I don't know. Keep in mind that these events are pulses, and pulses are less challenging than a high turbidity condition, which is persistent day in and day out. So those are 4 different scenarios of some of the data that I looked at that are case studies. They highlight different things and bring to mind different challenges that we have to consider when you think about turbidity. It's not only how high is the turbidity, it's how long does that situation last, that condition last over time. Dave, I've got another probably 5 minutes, so I could either stop right here. It's a nice clean stop and we can pick this up next month, or I could do another 5 minutes, up to you.
Another 5 minutes would be good.
Good. Yeah, I'd go another 5 minutes, Scott. And we do have a few questions to follow up with.
Okay. Okay, so going back to what can you do with turbidity data? What can it tell you about stream health? What can it tell you about stream restoration and problems in watersheds? So we just walked through a bunch of scenarios of how you can analyze turbidity values to compare against some biological threshold. And if you wanted to, you can compare those against a regulatory threshold if your state has one. There's another thing that we can do with turbidity data, and that's to work backwards from turbidity back to suspended sediment concentration and total suspended solids where we started from. And with those measurements, then we can dig into a much broader field of questions that get into figuring out where sediment comes from, how it's deposited, how it's transported, the effects of erosion, and that type of thing. So let's talk a little bit about the steps involved to get through that process. Many of you have, in conjunction with measuring turbidity at your sensor monitoring stations, you've not only been measuring turbidity, but you've been measuring suspended sediment concentration. And it turns out that if your suspended sediment in your stream is all one thing, if it was just clay or just silt particles or just sand, you could derive a really clean relationship between the signal that your turbidity sensor gives you and the concentration of that type of sediment. The problem is that you don't have just silt, clay, or sand. You've got a combination of all three of them. And each stream is different. And not only that, but those combinations are going to change over time in your stream. So yes, you can relate turbidity to suspended sediment concentration, as I'm about to explain more. But we need to be aware of the noise in that relationship. So if you've taken a grab sample of suspended sediment concentration or total suspended solids in a bottle, and at a point in time, you can plot for your stream this relationship, and you can fit a line through it, a regression line through it. In this case, it looks pretty good. The points fall pretty close to that regression line. What do you do with that? Well, you take that regression relationship and you can predict what suspended sediment concentration is for a given turbidity. Okay, so this allows you now for each turbidity value that you've measured, every 5 minutes at your stream, you can apply a simple equation and you can convert turbidity to suspended sediment concentration. The pattern looks the same, right? But the y-axis scale has changed. So now we've got a measure of suspended sediment concentration, kind of coming full circle to where we started today. If we want to calculate how much sediment goes downstream, we need to do more than that. We need to know how much discharge is going downstream. And in a very similar fashion, many of you have already done this. You've established the relationship between depth in your stream and discharge. So depth, level of water, discharge is mass of water moving per second. All right. And then here comes the magic part. Once you have suspended sediment concentration, and once you know discharge, you can multiply the black dot on the left by the corresponding blue dot on the right, and you get a suspended sediment load. So a mass per time that's going downstream past your sensor. And then if you add up and integrate all of that over time, in this particular case, it's 47,304 kilograms of sediment that went past your site. So this becomes a really powerful way to analyze suspended sediment load. But we need to be very careful because our regression relationships are not perfect. And if we include the predictive error in these relationships and we propagate that error through our equations, it becomes a little messier. We learned that that sediment load can vary by 35% because of the uncertainty in these regression relationships. So that 35% equates to 16,731 kilograms of sediment. So we've got a really big plus or minus around our calculation, even though we have some pretty good looking regression relationships. So that's a really powerful technique. Ultimately, that's in many cases what we'd like to do with our data, but we need to be very mindful of how much the error can creep in around our predictions. So that leads us to where we want to go next month, which is talking about how we can improve those turbidity measurements, ways that you can analyze your turbidity data. And really what we're going after here is not pushing one particular approach or agenda or question. It's all trying to give you the tools to better frame the question that you want to answer, and then help you through that process of getting to an endpoint where you're answering questions that you want to answer. And that's all I've got. Thank you.
Awesome, Scott. Wonderful presentation. Let's just get into questions right off. The first one was from Diane McGovern, I believe, if I can find that. Diane, what was your question? Let me see.
This is Diane. I didn't ask a
Sorry,
question.
no, I'm sorry. It was Heather Goss. I'm sorry, Diane. How do you Heather asked, how do you decide when the end of an event is for an analysis like this?
Good question. There are many ways that you can do that. You can do it visually, but that takes a long time if you've got 52 sites and 2 years of data to go through and pick out when do you start to see the peak go up and when do you stop. So we use different algorithms that automatically do that for us. And there are numerous ways to do it. I used an algorithm that picked out the peak and then bracketed that peak in the valleys between. So there's manual methods, there's automated methods and algorithms. There's a lot to talk about there.
You're pretty much just using the computer to find the turbidity humps.
That's right.
All right. So Fred Hartman was asking about the locations. We referred Fred to Monitor My Watershed. Fred also asked about plotting in Monitor My Watershed. Rachel, would you mind posting into the chat the link to the Monitor My Watershed help page? You can look at all kinds of data in there, Fred, and plot data, zoom in on axes, all kinds of stuff, overlay sites, overlay variables. And then we've got, let's see here. Okay, a couple of questions from Burton. How can we reliably sort out cause of turbidity to a stream upstream, that is upstream impact? And then number two is how can we effectively present data to local boards to get them to be aware of turbidity impact and what causes turbidity sediment?
Yeah, good questions. And those are at the root of a lot of what we want to do in many cases. Short of putting out thousands of turbidity sensors, probably the most effective way to start honing in on where turbidity comes from during storm events is to get out and walk the stream and take a windshield tour of the watershed during a flood event and see where you can find culvert pipes that you can visually see where dirty water might be coming from. I think, you know, it's a matter of, as Dave Bresler often says, of just getting familiar with your watershed, of mapping it to know where the tribs are, how they relate to one another, so that you can parse out, you know, which trib is upstream of brown water, basically, during floods. So getting out and walking the stream is one of the best ways to figure that out. How can you effectively present data to boards? First of all, I guess there's two parts there. The first is being confident in your turbidity data. Being confident that the turbidity data that you collect is coming from a sensor that's been routinely cared for, that you put your eyes on frequently, that you know that little optic window that we talk about so much when we talk about maintaining stations, making sure that's clean, making sure that when the water looks gin clear, that your turbidity sensor says that it's gin clear. That's where it starts, is making sure that you feel confident that that sensor makes sense to you, that the number that it's giving you makes sense. And then how do you put data in front of people? That's a great question, and it's probably worth an entire session here to talk through various ways to present data. Simply showing a peak during a storm downstream of something where you can point to a source in the watershed because you've seen it with your own eyes. Combining those pictures with a graph and arrows. Be creative, and I guess the bottom line is do what makes sense to you, that best represents your understanding of the situation, and make it visual.
Great, Scott. Tom Bruce had a question about the two turbidity levels on the stations. Tom, I did put in a response to that, but it's basically and Shannon, I don't know if you want to address this, but it's basically that at the low end, that is under 260, the two readings should be basically the same. And then the high one is the only one that goes above 260. Any other feedback on that, Shannon?
No, you're right. The sensor itself gives out two numbers, a low range and a high range. The low range goes from 0 to 250, and the high range goes from 0 to 1,000. And that's only so that the sensor could be 4 times more, give you 4 times better resolution in the low range. But since we already take high resolution readings, it's not really that necessary. But we report both numbers because that's what the sensor gives us, and it helps us in diagnosing the sensor. So if you ever see that the low and the high do not agree when it's below 250, then that should be an issue that the sensor or logger are having problems. But otherwise, if it's below 250, use the low range signal, and between 250 and 1,000, you use the high range signal.
And the first one is the low range?
Yes.
Okay, thanks.
And then Carol has a question. In your slides of the relationship of NTU to the development of caddis, rainbow trout, brook trout, et cetera, how long does the NTU remain at that threshold before the impact on growth and macroinverter index richness, for example, occurs? So how long are those levels persisting for that effect?
Right, and that's, I'm glad that you asked the question, Carol, because it emphasizes that whenever you talk about a threshold, you're probably also talking about a duration of exposure and an exposure regime. So it's great. The question highlights the importance of asking not only how high, but how long. And the answer is embedded within 5 different peer-reviewed articles that I can send you. And knowing Carol, you'll probably appreciate just getting the papers from me, if that would be all right. And everybody else is welcome to them as well. I can share them, but I'll send them to Carol if that's all right.
All right, so we've got questions piling up here. We're at 3:15. Carol has another one. Is the 35% error also common in professional measurements such as USGS? I missed if the error is due to lab measurement or due to the real changes in discharge.
No, the 35% is just what happened with that example data set that I pulled. If you get within 12%, you're doing pretty good. 12% error is pretty good, 10% error. So there's a ballpark to consider.
Okay, Yake has a question. Would there be value in being able to correlate the sediment generated in a given stream by the intensity of the rain event or runoff event? This approach produces a straight line with a high quality slope. So you're saying a relationship between discharge and turbidity.
Yeah, I would think so. Yes.
Yeah, and we've certainly seen those types of graphs produced. Here's Kevin Roth from Pennypack. Thanks for that data analysis. I am interested to see what the difference in TSS data for those two sites are before and after 11/26/2018. This is when a Toll Brothers development went up between the two sensor stations, and I reported them at least 38 times because of sediment leaving the site and discharging into the Pennypack. So there's another kind of sleuthing the landscape situation. So maybe that's the reason why Scott was seeing high levels at Paper Mill.
Yeah, and unfortunately, I can't remember what the time period was there, but that's a great.
You only looked at 18 and 19, right, Scott? You didn't look before 2018, right? So it was that time period.
Yeah, I don't know if I don't know if there was before data in that data set.
Okay, and, everyone note there.
2018.
Everyone note there also in the chat. Kevin put a link for a construction site monitoring workshop, so that's a nice reference that Kevin put there. Fred has a question, and we really should probably wrap up here soon. If dirty water seems offensive, what is being done? What can we do? What can we meaningfully appeal to regarding this stuff? Are there folks we need to support, to lobby, to beat on? Example, I'd love for the Chesapeake to be saved, a very big thing.
I think that's why we're all here, is to try to figure out not only where the dirty water comes from, but collectively what we can do. And it boils down to watershed management at so many levels and local decision making. That there's no silver bullet here. It's just a lot of grunt work on a lot of levels to figure out where the problems originate, to solve those problems where they exist, and to argue for stringent regulations where they make sense.
Great. Jim asks, are sharp drops in NTU at the Pennypack station, for example, due to cleaning the algae off the window? Generally, Jim, yes, those distinct drops are due to either cleaning, or if there was a big mass of leaves or something on there and it just pops off naturally, you'll often see those quick drops. Let's see. So we really should, the questions keep coming in. So Vicky has a question for Vicky Laubach. For regulatory purposes, which would you prefer, turbidity or TSS limits?
Great question. I don't have a good answer for that. Turbidity has advantages. Well, I said before that turbidity is this weird parameter with units that don't really mean anything. What is a nephelometric turbidity unit? Well, you can define it, but it doesn't make sense to anybody really. But it does make sense when you're talking about visual impairment of fish. So I think there are particular biological endpoints which may be more or less defined by turbidity or TSS. If you're interested in the flux of sediment, but definitely suspended sediment concentration is far superior to turbidity. If you're interested in how fish see, turbidity is preferable. So it depends. Hate to give that answer.
All right, so I think we should stop here. Great discussion. And if everybody wouldn't mind, we will do a Zoom poll now, the same one that we've been doing. So we'll take about a minute or so to do that. I'm going to launch it right now. So you should be viewing the questions. So we'll take a minute or so. Got folks responding.
Instant feedback can't be beat. This is great.
Not over 90%. We're at 91% right now. 94. $97. All right. This will wrap it up. Oh, wait, hold on. I got to take a picture. Great. All right. Okay, end polling. Excellent. Thanks so much, everyone. It's great. Excellent feedback. Okay, now I think because we're at 3:21, I think we should allow Christine Rogers to say a few words about what she wants to talk about. So go ahead, Christine.
Hi, everybody. If you recall from last month, Juniper and I were presenting about the executive summary that we've been working on with Dave Bressler to try to really get to the point of recommendations for the Paulin-Skill watershed. So we are now in the final draft version of that report. So we are going to be sharing it with all of you and would love your feedback. So you will be sent out after this meeting. And we really appreciate also the results of the survey that all of you were able to answer for us. The survey indicated that most of you are using your own data for engaging stakeholders locally and also your volunteers, that most of you want to identify and communicate the concerns with the general public. You all saw the value of the professional analysis of the data. And then also it was a high indication of support for displaying information graphically with recommendations. So the report that's going to be coming out is going to be specific to the Paulin-Skill watershed in Sussex County, New Jersey, but it's going to be a template that others can follow as we move forward and you look to do this own type of analysis for your own stations. So it'll be sent out and we'd appreciate any feedback or thoughts that you have on formatting, anything we're missing, just general thoughts. That would be extremely helpful. So thank you. And we appreciate your help in advance.
Wonderful. Thanks, Christine. And would you mind sending me the results of last month's surveys as well as the ones you're going to be sending out? Sounds
Yep.
like that's a lot
Absolutely.
of good information that would help us too. So that's excellent. Thanks so much. All right, so we have 7 minutes. So I'm going to share my screen and move on. We did the survey. So I'm just going to go into Stroud updates. And so the one thing, there's just a few ones I wanted to mention here. So Manage My Watershed. It's another another tool that's being launched in the Wiki Watershed Toolkit. There's a few pieces in there right now, stories. We're going to be loading a couple in there here in the next week or two from some of the folks that are on this call. George Seeds has written one, Carol Armstrong has written one, Francis Collins is preparing one. So there's going to be some really good information there. You know, it's broadly Manage My Watershed. The ones we're posting now are salt related primarily. So that is open to anyone that would like to do that. There's going to be a place for articles. There's going to be a place for resources, and there's going to be a place it's kind of like a forum where you can just ask questions. So feel free to folks can start making use of that now, right, Scott? Yeah, so good to go. And there will be some really nice stories coming in here that you can respond to, kind of like in a blog type situation. So another thing that I wanted to make sure to mention here is that there is a water quality project going on the Schuylkill right now from Reading to Philadelphia. It is being run by Schuylkill River Greenways located in Pottstown, and the technical aspects are being supported by Princeton Hydro with support from Stroud, John Jackson, and myself. And what Mike here from Princeton Hydro is hoping for is a bunch of volunteers to help with this particular component of this project, which is visual trash assessments. So they're trying to get folks to do this from anywhere from Pottstown down to Philly. It's a very simple protocol, visually based. You know, there's no training necessary. It's pretty much you just follow directions and you can do it wherever you like along the Schuylkill River from Reading down to Philadelphia. So just contact Mike Hartshorn at Princeton Hydro if you or anyone you know would like to be involved with that. They're really hoping for a lot of community support. So once again, we didn't get to this kind of tutorial section that we were hoping to. We're pretty much out of time right now. This presentation will be saved. What this tutorial is about is simply, I guess maybe just go here a little bit and just talk about it, but we're just talking about figuring out why your station has gone offline. So there's a legend here to see if it's gone offline on Monitor My Watershed. And you're essentially looking at battery levels. So if a station goes offline and the battery is higher than 3.5, then likely your cell board has malfunctioned. If the station goes offline and the battery is around this 3.5 level, then it's more likely that it's a actual power issue, and you may need to get more sun on your solar panel or just cycle the battery. But that's a super quick overview, but that's the gist of it. So feel free to go into this presentation once it's posted to here. So again, this for any new folks, this has all kinds of resources. This basically has all the resources that we have available. There's a lot in here, so you have to look, but here's where the monthly meeting materials are. So this recorded presentation will be in here as well as the actual presentation itself. So you can scroll through those slides I just described in more detail. So next meeting is Thursday, March 18th, 2:30 to 3:30. Sarah Damiano is going to do part 2 of the turbidity presentation, and then Patty Haug in April is going to talk about her monitoring of 5 Westchester stations in Westchester. We have no one booked yet for May, so if anyone is interested in talking about their work, feel free to be in touch. And feel free, you know, as I say in the emails, feel free to pass these meeting invitations on to others. Everyone is welcome. Just to point out again, haven't mentioned this in a while, there are mentors available. Carol, George, Rachel is available, and Krista's up in Bethlehem, PA. So she's further up north in the watershed. So anyone that's up north, you can call on Krista for assistance. And if there's anyone else that feels pretty experienced at this point and you'd like to be a mentor, feel free to be in touch. Okay. And that is everything. So we are at 3:29. So any follow-up questions or anything that we missed in the chat previously on questions? All good? All right. Thumbs up from Tom. Carol's waving, clapping from Francis.
I have a question, actually.
Oh, Carol.
But I'm sorry. It came from such one of the earlier slides. I forgot about it. Scott, when you were showing some of the standards for turbidity across states, wow, they were really hugely different. And it's hard to kind of understand that. So like Neshaminy only in Pennsylvania, I guess that was 100 NTU applies just to the Neshaminy. I mean, that's unthinkable. And then what was it, 50 or 40? And then meanwhile, Delaware is 10. It's hard to understand how there can be such differences. That doesn't apply to a lot of other things.
Yeah, Carol, and your comment emphasizes why turbidity is so tricky, because it depends on how long, it depends on how much, it depends on what it is. Remember, turbidity itself can be anything that bounces light off a particle. So is it necessarily bad? We don't know. So all of those questions compound to make it very difficult for the states to set a level. And notice that they're different for trout fishing waters, for warm water, for tidal water, for salt water. They can all have different levels. So turbidity is really complicated.
Scott, are there any other streams that you have evaluated for turbidity besides the ones you talked about today?
Dave, yeah, I looked at all of the streams that are on Monitor My Watershed. So there were, I think,
Okay,
55 within the DRWI.
right. I'll send you a summary of what I have, so at least you can have that as part of your background information.
Good, thank you.
All right, anything else? All right, well, thanks everyone. Really nice meeting today. We had a really good turnout, so hopefully it was valuable to everyone. We really appreciate all the great work you're doing, and you know, as usual, feel free to be in touch as needed. We're here to support you all. So take care and thanks everyone. Be safe out there.